Introduction. Welcome to today's lecture on the topic of medical nanotechnology, specifically focusing on the fascinating field of nanoscale drug delivery systems for pancreatic diseases. In this comprehensive script, we will explore the anatomy and physiology of the pancreas, as well as the challenges in drug delivery to this vital organ. We will delve into the various nanocarriers used for pancreatic drug delivery, the mechanisms of drug release in nanoscale drug delivery systems, and the strategies employed to target drugs specifically to the pancreas. Additionally, we will discuss imaging techniques to evaluate drug delivery, the importance of nanotoxicology, the development of bio-inspired drug delivery systems, the translation of nanoscale drug delivery to clinical practice, the role of personalized medicine, and finally, future directions in this evolving field. Let us now begin with our first section. Nanoscale drug delivery systems refer to the application of nanotechnology in the design and development of drug delivery platforms at the molecular or nanoparticle level. These systems are specifically designed to improve the therapeutic efficacy of drugs by enhancing drug stability, targeting drug delivery to the desired site, and controlling drug release kinetics. Nanoparticles, liposomes, micelles, dendrimers, and other nanocarriers are commonly employed in nanoscale drug delivery systems. The advantages of these systems include increased drug solubility, prolonged drug circulation time, reduced side effects, and improved patient compliance. The pancreas is a vital organ located in the upper abdomen, behind the stomach. It plays a crucial role in digestion and glucose metabolism. The pancreas consists of two main functional components, the exocrine pancreas and the endocrine pancreas. The exocrine pancreas secretes digestive enzymes and bicarbonate into the small intestine, aiding in the breakdown of carbohydrates, fats, and proteins. The endocrine pancreas, which includes the islets of Langerhans, produces and releases hormones such as insulin and glucagon, which regulate blood glucose levels. Understanding the anatomy and physiology of the pancreas is essential for efficient drug delivery and targeting. Pancreatic diseases, such as pancreatitis, pancreatic cancer, and diabetes, pose significant challenges in drug delivery due to the unique characteristics of the pancreas. The dense fibrotic stroma surrounding pancreatic tumors hinders drug penetration, while the enzymatic environment of the pancreas may degrade therapeutic agents. Furthermore, the selective vulnerability of pancreatic beta cells in diabetes necessitates targeted drug delivery. Overcoming these challenges requires the development of innovative drug delivery systems specifically tailored for pancreatic diseases. Nanocarriers have revolutionized drug delivery by enabling controlled release, enhanced drug stability, and targeted delivery. In the context of pancreatic diseases, various nanocarriers such as liposomes, polymer nanoparticles, and inorganic nanoparticles have been investigated. Liposomes, for instance, can encapsulate hydrophilic and lipophilic drugs, protect them from degradation, and improve their bioavailability. Polymer nanoparticles, on the other hand, offer tunable properties, allowing for precise control over drug release and targeting. Inorganic nanoparticles, such as gold nanoparticles, have unique optical properties that can be exploited for imaging and therapy. The mechanisms governing drug release from nanoscale drug delivery systems are critical in determining therapeutic efficacy. Common drug release mechanisms include diffusion, erosion, and stimuli-responsive release. Diffusion relies on the concentration gradient, allowing drugs to slowly diffuse out of the carrier. Erosion involves the degradation of the carrier material, resulting in drug release. Stimuli-responsive release relies on external triggers, such as pH, temperature, light, or enzymes, to trigger drug release. Understanding these mechanisms is crucial for designing drug delivery systems with optimal release kinetics. Targeted drug delivery aims to selectively deliver drugs to the pancreas, minimizing off-target effects. Passive targeting exploits the enhanced permeability and retention effect of nanoparticles in tumors, allowing for increased accumulation. Active targeting, on the other hand, employs ligands, antibodies, or peptides that specifically recognize surface receptors or biomarkers present on pancreatic cells. Targeting strategies can enhance drug delivery efficiency and reduce systemic toxicity, improving the overall therapeutic outcomes for pancreatic diseases. Imaging techniques play a crucial role in evaluating the effectiveness of nanoscale drug delivery to the pancreas. Non-invasive imaging modalities, such as magnetic resonance imaging, MRI, computed tomography, CT, and positron emission tomography, PET, allow for real-time monitoring of drug distribution, release, and tumor targeting. Optical imaging techniques, such as fluorescence imaging and bioluminescence imaging, offer high resolution and sensitivity for visualizing drug delivery at the cellular and molecular levels. These imaging techniques enable researchers and clinicians to optimize drug delivery strategies and assess treatment response. The potential toxicity of nanomaterials used in drug delivery systems must be carefully evaluated to ensure patient safety. Nanotoxicology involves studying the adverse effects of nanoparticles on various biological systems. The physicochemical properties, such as size, shape, surface charge, and composition, influence the interactions between nanoparticles and biological entities. Understanding the biocompatibility and potential toxicities associated with nanocarriers is essential for developing safe and effective pancreatic drug delivery systems. Bio-inspired drug delivery systems aim to mimic natural biological processes and structures to improve drug delivery to the pancreas. For example, biomimetic nanoparticles can be engineered to resemble cell membranes, enhancing their stability, biocompatibility, and targeting capabilities. Additionally, exploiting physiological pathways, such as receptor-mediated endocytosis, can facilitate efficient drug internalization and release. Bio-inspired drug delivery systems hold great promise for overcoming the challenges associated with pancreatic diseases. The translation of nanoscale drug delivery systems for pancreatic diseases from the lab to clinical practice is a complex process that involves regulatory approval, safety assessments, and clinical trials. Collaborations between researchers, clinicians, and pharmaceutical companies are crucial for advancing these technologies. Overcoming the hurdles associated with manufacturing, scalability, and cost-effectiveness is essential for widespread adoption and successful integration of nanoscale drug delivery systems into clinical practice. Personalized medicine, also known as precision medicine, focuses on tailoring medical treatments to individual patients based on their unique characteristics, including genetic makeup, lifestyle, and environmental factors. In the context of pancreatic diseases, personalized medicine holds great potential for improving treatment outcomes. Nanoscale drug delivery systems can be customized to deliver drugs at precise doses, frequencies, and routes, optimizing therapeutic responses while minimizing side effects. The integration of nanotechnology and personalized medicine may revolutionize the management of pancreatic diseases in the future. The field of nanoscale drug delivery to the pancreas is continuously evolving, with many exciting developments on the horizon. Researchers are exploring innovative drug delivery approaches, such as cell-based delivery systems, extracellular vesicles, and nanorobotics. Additionally, advancements in imaging techniques, artificial intelligence, and nanosensors are expected to enhance diagnostic and therapeutic capabilities. The integration of nanotechnology with other emerging fields, such as gene therapy and regenerative medicine, may pave the way for groundbreaking treatments for pancreatic diseases. In conclusion, nanoscale drug delivery systems offer immense potential for addressing the challenges associated with pancreatic diseases. The unique properties of nanocarriers, combined with targeted drug delivery strategies, imaging techniques, and personalized medicine, have the power to revolutionize the way we diagnose and treat these complex conditions. As we continue to unravel the mysteries of the pancreas and exploit the potential of nanotechnology, we move closer to unlocking new therapies and improving the lives of millions of patients worldwide.